Okay, guys, so let's get started. I'm turning off my video. My internet connection is not very stable, so this will help with that. Okay, so welcome to week two of bioinformatics algorithms and applications. Last week we talked about some introductory stuff about bioinformatics, where it is used, different types of databases that we can access, and so on. So today's topics are mostly focused on proteins and peptides, their databases, and uh, yeah, how we get the data and so on. Yeah. So just to give you a very quick overview about proteins. So what are proteins? Proteins are basically polypeptide chains of uh, polypeptides, right? So you have these amino acids. We have 20 amino acids as we discussed in the last class. So we have the codons, three nucleotide codons that code for a particular amino acid, and that codon is a degenerate codon, so multiple codons can code for one amino acid. So out of 64 combinations, we have 20 essential amino acids that are coded by our genes, right? So a protein is basically a long chain of these amino acids bonded up one on next to the other by peptide bonds. These are like one after another. We have a sequence of these amino acids that form the protein, which is the polypeptide or the polypeptide chains. The peptide bond that connects two amino acids in a polypeptide chain is a very interesting bond in itself. So this is basically, I'll just quickly show you. So this is basically the amino group, right? Uh, So this part that you see is basically the amino group where you have so this is your COO minus, which is your carboxyl group. Then you have another CH here. This is the alpha carbon, the carbon that is directly attached to the COO group. And to this carbon, there is one amino group attached, which is the NH3 plus group, and one hydrogen, and the R2 is basically a long chain of other carbons that can be any number of carbons. So depending on the structure of R or the composition of R, we have this 20 different amino acids. But this is the general structure of how the amino acid looks. And if you connect, so both of them are similar. If you have different R1 and R2, you'll have different amino acids, right? Now, when you join this to amino acid, this bond is formed, which is the peptide bond. So the CO on the left, this has a negative charge, and the amino group on the right, this has a positive charge. So they join together to form a peptide bond. And generally, there is, since these R groups, R1, R2, these are very large groups of carbon, hydrogen, and other compounds. So these are like large groups. So these form steric hindrances. Like the rotation, just keep going back to class 12 chemistry, the rotation that is possible between in this bond is severely restricted because this is a single bond, so rotation is freely available. If it's a double or a triple bond, rotation would not have been possible. But since this is a single bond, rotation is technically possible. But since this R1 and R2 are very huge and complicated groups, it brings in steric hindrance along with it. So you cannot just rotate along the bond of this peptide bond. Right. So generally, it forms and kind of anti-parallel kind of a strand where you have the R groups from each amino acid which orients opposite to each other in the polypeptide chain. Right. So coming to different amino acids, so these are your basically all your 20 amino acids that are possible. You see some of them have a lot of these aromatic groups, which are your benzene rings, basically. Depending upon the number and the type of the ring that is present, we have basically three amino acids which are aromatic in nature. So this ring structure brings the aromatic characteristic of the particular amino acid. So tyrosine, tryptophan, and phenylalanine are aromatic amino acids because of the presence of that particular those particular ring structures. So as I was saying, the R group in this case, as you can correctly imagine, so this is NH3, this is COO, this part is the R group. In this case, this part, in this case, the ring structures and so on. So the all the compounds that are marked in light and dark blue are your basically amino groups, and the rest of them are your big R groups that give the amino acids their characteristics. Uh, does anybody want to say anything? Okay, I'm muting you because it brings about disturbance. So yeah, if you have anything to ask, please do so freely. Yeah. So yeah, on the left hand side you have these set of amino acid structures, and on the right hand side you have this is a table basically. A lot of stuff is given here: molecular weight, your molecular formula, and residue weight, the dissociation coefficients, and all of this. We're not going to go into so much of details, but yeah, each of the amino acid is very specific in terms of its chemical properties, and it is very distinct if you compare to other amino acids, but of different groups. So these are, as I was saying, the R groups here, the ones in purple, they determine what kind of amino acid it is going to be. So if it's a ring-like structure, uh, you have aromatic amino acids. So the R group in these amino acids are hydrophobic side chains. These include phenylalanine, tyrosine, and tryptophan. These three particular ones which have this a benzene ring like structure which gives them the aromatic properties. Then you have polar amino acids, the uncharged R groups. In this case, the amino acids are uncharged and they're more polar than hydrophobic amino acids. And this includes your charged amino acids like serine, threonine, cysteine, asparagine, glutamine. So serine and threonine, I think you'll come across in a lot of biological pathways because these are the regions where these are since these are charged amino acids, these are the regions in the protein where different modifications can happen. So kinase basically is an enzyme that adds phosphate groups to your compound, right? So kinase would typically there are a lot of kinase that acts on these serine threonine residues. So if you have charged residues on the polypeptide chain, it is easier to add modifications to those regions of the protein. One second. Mm, Okay. Yeah. Moving on, then you have positively charged R groups where you have these are acidic amino acids which take which with a net negative charge, and they actually attract positive groups to them, and these properties of amino acids being charged, them being aromatic, or them having a long heavy side chain, these all factors impact how the polypeptide chain, the protein that is getting formed by these amino acids, are going to fold. The secondary and the tertiary structures that are going to be formed from this long chain of polypeptides is determined by the properties that are of the amino acids that are present in the specific locations, right? Uh, yeah. Okay, so as I was saying, so you have a long polypeptide chain. Now, this chain of polypeptide is not what is found inside the cell. Inside the cell, the protein is found in a much more complicated structure. So we have this hierarchy of protein structure starting from just a long chain of amino acid, which is the polypeptide chain, which gives you the primary structure of the amino acid, which is the sum of all protein structural elements arising from the covalent bonds. The secondary structure, we'll discuss these in a little bit more detail further down the line. The secondary structure is basically polypeptide strand structures dictated by hydrogen bonding between the carbonyl O and the amido H groups of the polypeptides of the peptide bonds. I'll tell you why the double or the single bond is important, because as you saw here, this this is basically one single bond and one double bond, but this dotted line is given because there is resonance in this structure, because one single bond is and one double bond will give you a kind of a uh, one and a half kind of a bond like thing, because of the resonance that is happening chemically in that particular region. So that is also important, because if you might imagine that one side of it is a double bonded with O and the other side of it is a single bond, right? With the single bond side might be able to freely rotate, but that is not the case, because there is resonance in the structure, and that is not a like a simple single bond. This is almost a double bond formed by resonance. So those kind of things become important when you're folding the protein and making it in making secondary structures out of the protein. After that, you have tertiary structures, which is basically the arrangement of these secondary structures into larger three-dimensional architectures. And finally, the quaternary structure. Not all proteins have quaternary structures, but a lot of them do. It is basically the non-covalent association of multiple protein subunits into a large multimeric complex. We'll talk about a few examples from each of these structures in the further slides. Okay, so coming to the primary structure, as I was saying, it is basically the long list of long polypeptide chain of amino acids. So the things that we are concerned about in the primary structure are the sequence of the amino acids, the disulfide bonds, because there is a lot of the cysteine. Cysteine is basically an amino acid which has this sulfide bond. So these disulfide bonds provide structure to to these amino acids as well. So these bonds also are very important. You have we are concerned with proteolytic cleavages, where you have proteolytic enzymes that kind of break these peptide bonds between different amino acids, and also post-translational modifications where so if you remember, the information flows from DNA to RNA to protein, right? So from DNA we make mRNA through transcription, and from mRNA we make protein through translation. Now, after translation has happened inside eukaryotic cells, and as well as us humans, because we are also eukaryotic, we have a lot of post-translational pathways that get activated, and a lot of modifications are put on these proteins that are formed after translation. So these can be signaling molecules. So in some cases, they add ubiquitin to the protein. So this signals the cell that this protein is malformed or like ill-formed, and we need to destroy this protein. So if you add a ubiquitin tag to that protein, the cell will recognize it, and it will get packed into Golgi bodies, and then all the ubiquitination process will happen and the protein will be broken down. So there can be other modifications as well. So if you have a poly A tail in the mRNA, that's like a post-transcriptional modification. And again, at the DNA level also, you have DNA modification. So at every level of this information flow, we add modifications, not we, the cell adds modifications to these structures or these compounds to signal or mark the compound for a particular function or a particular particular pathway or anything like that. So we are going to be there's a lot of things that can happen immediately after the protein has been formed. So transcription, post-translational modifications are one of them. Um, and yeah, so this is how a protein structure is represented. On the left, you have the amino terminal residue. In the middle, you have these peptide bonds, and on the right, you have the carboxyl terminal residue. So 5 prime to 3 prime. We do not call 5 prime to 3 prime in amino acids because there's no 5 prime and 3 prime technically. But like if you want to carry over the nomenclature from replication and transcription, basically it moves from the amino terminal residue to the carboxyl terminal residue. Hmm. Okay. Okay. Now coming to the secondary structure. Uh, yeah. Now, coming to the secondary structure, as I was saying, the resonance between this particular bond is very interesting. So you have a double bond here and you have a amino group here. So this double bond actually has a resonance kind of a structure that kind of shortens this particular carbon nitrogen bond, and it also hinders rotation along this particular peptide bond as well. So yeah, the peptide bond, although it looks that it has one single bond, one double bond and one single bond, the single bond, it is not very flexible, it is rigid and it is planar. Right? As I was saying, you have steric hindrance of these of the big R groups that are giving configuration to these amino acids. This provides steric hindrance, and it also limits the rotation of this particular bond, which is a particular carbon atom, the bond along this particular carbon atom, which is the alpha carbon, as we say. And depending on the possibilities of rotation, we have the Ramachandran plot. We'll come to talk about the Ramachandran plot in details in the next weeks. But yeah, the secondary structure is determined. It is not as free as a primary structure, as you can understand, because this is a folded kind of a structure. Because after the peptide bonds have been formed, it forms a rigid planar structure. So it is not very it is not very free flowing or like very flexible in nature. Uh, so alpha helix is basically one type of secondary structure. Basically, there are two types. One is the alpha helix and the second is the beta sheet. So it basically the alpha helix is basically forms a right-handed helix, which is these are the details of it with uh, 5.4 angstrom as the pitch of the helix and 3.6 amino acids per turn, and this is how the helix looks like, this helical structure. And the side chains, the big R groups, they actually extend away from this helical structure, which is understandable because if they are pointing towards the inside of the helix, it becomes sterically not very feasible to have this kind of a structure, right? So yeah, this is what the alpha helix looks like. And there is also beta sheets, which are uh, basically sheet-like structures which form anti-parallel streams. As you can see, this example is showing that the big groups actually are facing each other. So you have these anti-parallel strands. In the beta sheet, the hydrogen bond acceptor is always in a different strand than the donor, which forms. So these are sheets, like so. These are not peptide bonds. These are peptide bonds here, right? These are peptide bonds here. So in the beta sheets, you have bonds forming between two strands of of a particular protein. So this is basically a folded structure here, and inside these, the formation of these bonds actually keeps this particular structure stable. And uh, yeah, so mostly it is anti-parallel, but you can also have parallel strands as well. But it is thermodynamically less feasible than the anti-parallel because in antiparallel, the steric hindrance and is lower, and the bond formation capability. Uh, uh, the bond formation capability here is much higher in case of antiparallel strands. Okay, now we talked about primary structures. Secondary structure, we talked about a little bit about alpha helix and beta sheets. And then we come to tertiary structures. Just a second. Guys, a small request, if you can join in time, that would be good because every time I have to like go back to the screen and admit people, it causes a little bit of distraction for me. So it will be good if you can join in time. Right? Okay. So in tertiary structure, so it basically forms the whole 3D structure of the protein. So these are like, if you see, if you isolate a protein in lab and you try to do, say, for example, X-ray crystallography on it and try to see what is the overall structure of that particular protein, mostly you see a very few classes of these proteins. They are either globular or fibrous and composed of virtually any combination of secondary structures. So there can be like n number of alpha helix, n number of beta sheets, or there can be neither of either of them. So it's a very like a lot of combinations can be possible. Now, globular proteins, they are not very elongated and they are shaped like a glob, a ball-like structure. And these are most non-structural proteins, including enzymes and other proteins are globular in nature. So if you can imagine, why do enzymes have to be globular in nature? If you know about how enzymes work, so enzymes have a active site inside their inside their protein, which is a small residue of a few amino acids, which is actually where the substrate goes and binds to the enzyme, and the catalytic process happens after the substrate goes and binds to the amino acid. Now, that kind of a structure that you have to have, where you have a small set of amino acids where the substrate will come and dock, and you have to cover that whole thing up. So globular structure is very efficient for that, right? So fibrillar structure is not very good for that because it's a long fiber. But in case of enzymes, you need a more of a globular structure. Fibrous proteins, on the other hand, have very elongated structures. They are typically major constituents of larger physical structures. Example, keratin, collagen. Collagen is a very good example of these long fibrous proteins, and how actually the this long structure helps in muscle movement. So proteins like actin and myosin and the other proteins that are involved in motor movement are actually very optimized to be to work in that situation, and this long fibrillar structure actually helps them a lot in terms of that. So this is how the tertiary structures look like. So this is basically a tertiary structure of an enzyme called lysozyme, which is present in, if I'm not wrong, human tears. If I'm not wrong, this is an antibacterial. Uh, let me check. I've forgotten what lysozyme comes from. Uh, yeah. Lysozyme is present in tears, saliva, and mucus. So this is kind of a antimicrobial enzyme that acts as a first line of defense of the innate immune system. And so this is how it looks like. So if you see, this is a globular protein. Why is it a globular protein? Because it needs to bind to something. It needs to bind to the microbe. It needs to bind to the receptor the of the bacterial cell wall or whatever. So where that kind of binding is required, a globular structure helps to do that. So this is how the tertiary structure of the enzyme lysozyme looks. Then you have proteins like keratins, where you have this long stranded alpha helical coiled structure, and so defects. One of the major defect in keratin proteins actually cause different types of keratinopathies, and there are a lot of mutations that can happen. So this is one example of a disease, which is epidermolysis bulbosa simplex, which is caused by this particular mutation in K14 protein. So again, these are very this type of long proteins are very uh, long fibrous proteins are also very important, and they have different kind of roles other than globular proteins as well, but equally important and uh, very important for survival of human beings, specifically movement type uh, functions, right? And then also this is basically another fibrous protein, which is collagen. Again, a very interesting thing in collagen is basically every third amino acid is glycine, and proline content is also very high. So if you have glycine and proline, if you yeah, if you go back here, where is glycine? Yeah, this is glycine. You can see proline. Huh. Here is proline and here is glycine. So these are like small amino acids. These are like kind of the smallest amino acids that are possible. Proline is the, I think glycine is the smallest, if I'm not wrong. Yeah, glycine is the smallest, but proline is the second smallest. So in when you are forming globular structures, if you have very high number of complicated long chain amino acids, the folding does not happen very well. So if you have these small amino acid residues, these can be folded very easily. So again, this is also very interesting. If you want to know more about it, there are a lot of resources, a lot of studies done on collagen proteins. You can look it up. It's quite interesting, right? Now we come to the quaternary structure. These are basically uh, if you have two set of tertiary proteins that are basically subunits of a larger protein, then these two tertiary structures will come together to form a quaternary structure, which will form the final protein. Uh, uh, so as I was saying, if multiple of these tertiary structures come and make the quaternary structure, these quaternary proteins can be mostly homomeric or heteromeric. So, what do you mean by homomeric? Basically, if there are n subunits of that particular protein, all the n subunits are of similar nature. But in on a lot of cases, you have a multimeric protein. A lot of different subunits of proteins come together and combine and form a different protein. So that is multimeric. So you have different subunits of protein that are forming the larger one. And this association of multiple subunits confers capacity for sophisticated regulation of activity, which is allosterism. I don't know if you're familiar with allosterism or not. Is basically if there are if there is an active site and there is another site inside the enzyme, in that site another protein comes and bind it, and it kind of changes the conformation of the protein itself and opens up the active site. So if that particular protein did not go and bind to the other site, the active site won't be opened. So this is a very sophisticated way of regulating enzyme catalysis inside the cell. So this is possible because of quaternary structures of proteins that are there. And association of enzymes catalyzing sequential reactions into multi-enzyme complexes accelerates flux of metabolites through pathways as well. And so this is how hemoglobin looks like. So these are different subunits of the pink one is a different subunit, the yellow one is a different submit, and this is the PDB ID of this particular protein, which is hemoglobin. You can just go to PDB and search this particular ID, and it'll bring up the structure, and you can go through the structure. So we have discussed amino acids. We have discussed how amino acids can join and form primary structures, how you can form secondary structures on top of primary structures, then tertiary structures, and multiple proteins of these structures, tertiary structures can come together and form quaternary structures in complicated proteins like hemoglobin. Right? Okay, sorry. Now coming to functions, as we all know, I don't think I need to talk too much about functions of proteins because I think a lot of you have a biological background and you're very familiar with like all the different kinds of things proteins does. But I just want to give you a kind of a overview. So the first important thing, function of protein is acting as an enzyme, right? That is one of the major function of protein inside the cell. Um, they act as catalysts that enhance the rate of these chemical reactions in various pathways, not only metabolic. Here it is written metabolic, but it can be other pathways as well. The fibrous proteins are component of various tissues holding skeletal elements together, such as collagen, which is a structural subunit of connective tissue. As I was talking about, movement functions are dependent on fibrous proteins like these. Proteins serve as carrier of genetic characters and hence govern inheritance of traits. This is also a very important class of proteins, which are basically uh, proteins that are present inside the nucleus. These can be transcription factors, which actually go and bind to the DNA and tell the DNA to transcribe itself, and so on and so forth. So these are another form of inheritance, which uh, regulate different forms of inheritance of different forms of traits depending on the gene regulation. 1 second. Ah, sorry for that. Okay. Uh, right, where was I? Yeah. So nucleoproteins. Then you have proteins which perform transport, right? So there can be a very good example of that is the F0 F1 ATP synthase. I'm sure a lot of you might be familiar with that protein. So it is a protein that is present inside the inner mitochondrial membrane. It is heavily involved in electron transport chain pathway of the cell, and this is a protein which acts as a hydrogen pump, right? So this is a pump-like protein. My camera, I'll turn my camera on for this. Yeah, yeah. So you have like a lot of subunits, but you can imagine it as like a two major subunit protein sitting on top of one another, and they can rotate freely on opposite sides. So that is a very important quaternary structure of the protein. So that particular enzyme, it's F0F1 ATP synthase, acts as a hydrogen pump, which transports hydrogen ions from an area of lower concentration to higher concentration. You are transporting from an area of lower concentration to higher concentration, you need energy, you need pump. So that is why the name ATP, hydrogen pump, and the energy is given by ATP molecules. So a lot of transport happens inside cells. Some of them require energy, like this particular one, hydrogen pump. Some of them do not. Some of them are just channels inside membranes where it allows diffusion of molecules from an area of higher concentration to an area of lower concentration. There can be transport proteins. There can be proteins that help tag a particular protein, and as I was saying, the example of ubiquitin, it goes and tags a particular protein, and that tag is recognized by the cell to kind of kill that protein or destroy that protein. So these kind of things also are very, these kind of functions actually are very important for proteins, right? So depending on if energy is required or not required, you can have active transport, which is if energy is required, that is active transport. If energy is not required, we call that passive transport, right? So other than that, you have protein hormones that regulate growth of different plants, animals, etc., etc. Your blood plasma has a lot of soluble proteins, blood clotting proteins like thrombin, etc., which help in blood clotting when there is a cut on the skin. And there can be other signaling molecules as well, like interferons. So these are regulatory glycoproteins produced by these eukaryotic cells in response to virus infection, endotoxins, antigenic stimuli, and many parasitic organisms. So these interferons act as signaling molecules, kind of similar to hormones, right? So, and also uh, uh, like some of the proteins might be antibiotic in nature as well, which can kill harmful pathogenic bacteria and other small microorganisms. So this is just a slide that wants to tell you about the level of functions the proteins are involved in. From any kind of functions you can think of, the proteins are involved in that. So it's very important that we learn about them and their structures and how actually these things help the protein function the way it is. Okay. Okay. In bioinformatics, at different levels of these structures, there are a lot of studies that try to predict. So primary structure is quite simplified. It's just a sequence of amino acid. You don't need to predict anything from that. But from secondary, tertiary, and quaternary structure, if you're just given the sequence of amino acid, can you predict the secondary or the quaternary structure of the protein? It turns out, in some cases you can do, but in a lot of cases you can't, and it is a very, very difficult and an open-ended question, which got solved in not I won't say it got solved, because we still am not we still are not very sure about like systematically how and what are the things that actually help the protein make that particular structure. But using different deep learning models and machine learning models, we can predict if this is a sequence, this is how the structure is going to be. I think AlphaFold, from Google DeepMind, solved this problem a few years back, and I think the DeepMind CEO got a Nobel Prize for that as well. So it's a very open-ended and a very important biological question that has been there for the past 50 years. So using chemistry, yet we are still used to solve it, but using machine learning and deep learning, we can at least predict that these are the structures that are going to be from a particular sequence. Now, the basic level of studying proteins in bioinformatics is just comparison of of the sequence of the proteins itself. So what are the sequence of the amino acids present in a protein versus what is the sequence of the amino acid present in another protein? So can the differences in these sequence tell us about the functions or the characteristics of the proteins itself? Other things can also be inferred, like evolutionary relationship. Say, for example, I'll tell you one interesting example is of histone. So histone is a protein that is present inside the nucleus, and it's an octamer. It's a octamer protein which has 8 subunits, and it's a positively charged protein along which the negatively charged DNA is bound. Right now, histone proteins, interestingly, if you look at different organisms, it is one of the most conserved proteins in uh, across different organisms, right? So there, if you compare different proteins from different organisms, same proteins from different organisms, and you find certain differences in those protein sequences, this can tell you a lot about the evolutionary constant, about that how evolutionary forces have worked on it to mutate that to become something else from its ancestor, right? So that becomes very important. So if if two proteins, say for example, another important example would be like if you have a protein that you do not know what it is, like you have isolated a protein from an organism, you don't know what protein it is and what its function is or what it is doing. But what you know is the sequence of the amino acids in that protein that you can easily find out using there are techniques like mass spectrometry nowadays which you can use to find out the sequence of the protein. Now, once you have found out the sequence of the protein, what you can do is try to match that sequence to other organism proteins. So, for example, you have you have isolated a protein from a chimpanzee. It matches very closely to, say, for example, the actin protein. Or the collagen protein inside the human cell. So that means that the protein that you have isolated is very close in function or as well, because the sequence is very similar. So you can safely say it's very close to function to the collagen protein in the humans because the sequence are very similar. And if you have similar sequences, it tells an evolutionary relationship between the individuals as well. So for example, a chimpanzee protein A and the human protein A will be very similar in sequence. And it tells you about the evolutionary relationship. And by extension, if there is a similar sequence, there is an evolutionary relationship, and there is also related function because sequence is similar. Uh, and there are some proteins which are called as orthologs to one another, which are basically almost same function, but they are present in different organisms with slight different sequences. So why I'm telling you all this is because studying the sequence of the protein at that level can give you a lot of information across species, within species as well. So this is the first level of study that bioinformatics does, is basically comparing protein sequences between organisms, across organisms. It can be anything depending on your research question. Right. Now, one such way to do it, one technique to ascertain sequence similarity is by using dot plots. So, for example, you are given two sequence of length n and m respectively, and you are asked, do they share a similarity? And if so, which region is the similar region? So in dot plot, what they do is you make a n by m matrix. So your n is basically your rows will represent your protein A, and your columns will represent protein B. Right. Uh, that's the n by m matrix. Now, inside the matrix, the values will be determined if at position 1 and so at position J of protein A and at position J of protein B, is there a same amino acid? If that is the same, you put a 1. Otherwise, you put something else. We'll talk about that, what we will put if there is a mismatch. There can be different types of mismatches, but yeah, if it's a match, you put a 1. If there's a mismatch, you put some penalty on it, and you put a score like that. So that is how a matrix of that sort is made. And if you print that graphically, it looks something like this. So this is a more of a deeper level, sorry, a more of a zoomed in version of a dot plot. So these are not amino acid sequence. These are technically DNA sequences, but you can do the same for amino acid sequences as well. There's not much difference in terms of like what is the penalty you are going to give if there are sequence mismatches. That will be relevant if you are doing protein comparisons versus DNA comparisons, because what kind of uh, changes have occurred, depending on that you'll put a score on that. But other than that, the idea is very similar. That, see, in this position, in the third position of, let us call this protein A and this is protein B. So the third position of protein A matches the first position of protein B, so you have a dot here. And if it does not match, you don't have dots here. So diagonals from top left to bottom right correspond to the regions that are identical in both sequences, and the diagonals in the perpendicular direction correspond to reverse matches, which tells you about inverted sequences. So a sequence, so for example, if you have a sequence here, CAT, and if you have something here, TCA, so you see it's a perpendicular relationship here. So if you have an inversion kind of a thing, it'll go perpendicular. If it's a full match, it'll go on the leading diagonal. Right. So this is how a dot plot would look like if there were uh, uh, the relationship between these set of sequences of protein A versus protein B is not very clear. It will be a haphazard kind of a set of lines here. You cannot infer anything from this. And in an obvious case, what would you see is a straight line like this here in the leading diagonal. So if it's a complete match, you'll have straight leading line like this. Sorry, leading like line like this. If not, you'll have the scattered points like these that is happening. So I'll talk mostly about like this dot plots. I'm going to talk in with respect to genomic sequences, but as you can understand, there is not much difference if you want to do it with a protein sequence. It's kind of the same. It's just that your scoring patterns will differ. So in this case, in for DNA, we
Sir,
yeah.
I have a question. In the previous image that you have shown, so there were small diagonals all over the plot, right?
Yes, yes,
As you can see. So
yes.
this means that some nucleotide sequences, like 3 to 4 nucleotide sequences, matching repeatedly in the
Yeah,
in the
Yeah, yeah,
protein.
definitely, definitely. So
So,
what?
so that's why isn't it whole as a whole, if you look at it, isn't it similar to each other? How it,
No, I'll tell you why. So, for example, let us assume there are four letters here, ATGC, and there is some other four letters here, right? So, what you are seeing here is, if you see this, let me zoom in here. So, here, as you can see, there is a diagonal line here, right? And there is also a diagonal line here. So what it means is this region of this particular protein matches with this region of that particular of the second protein. And if it's safe to assume that those are similar positions. So for example, say position 345 matches with position 345 here. For that, you have gotten this particular diagonal, right? That is fine. Now again, this particular region 345 also matches with somewhere here, say 789. So the matches are across. So the whole protein, if you break it down into smaller nucleotide sequences, sorry, smaller polypeptide sequences, for a set of like say three or four set of sequences together, those three or four set of sequences match with a lot of the regions in the other protein. Like it's not a one-to-one match. So if it would have been a one-to-one match, so protein 345 here would match to only protein 345 here, and you won't have another parallel line here. This parallel line here or the other parallel lines that are in and around this match is telling you that that region of protein A is found in multiple regions of protein B. So it's not a one-to-one match, right? There are matches. There will obviously be matches because you have 20 amino acid sequences in case of proteins, and you have four letters possible in case of DNA. And there are evolutionary wise, a certain sequence is always more frequent than others, right? So there will be a lot of matches, but is it a complete match? Is it a one-to-one match? In this case, it's not because you see along this particular diagonal, you have so if you assume these are like amino acids 4, 5, and 6, they occur here, which is basically 2, 3, 4 in protein A, protein B, sorry. They occur here, say for example, 4, 5, 6 in protein B. They also occur here, 7, 8, 9 in protein B. So this is not a one-to-one match. It's like that set of amino acids is present all across the protein. So this is not a good match at all. So have I
yes,
be
sir.
able to make it clear?
Yes, sir. Yes, sir. I understood.
Yeah, okay. Thank you. Uh, so if you have a clean line on the leading diagonal, if there will be some offsets, there will be some parallels like this because some portion of protein A will always be in multiple regions of protein B. That can be possible. But if it is like that is the only signature that you're getting, that means it's not a good match. The good match would be you have a good dark diagonal line, and then you can have some parallels of those lines somewhere in the plot. That is possible. But the leading diagonal line should be very prominent if that's a good match. Okay. Yeah, as I was going about, so this here the example I'm talking about is for genomic data, but again, it's going to be very similar for protein data as well because it's just a sequence. So each dot will correspond to a pair of orthologous. In this case, we are matching genes. And the key features of these plots is the distinct X-shaped pattern. These suggest that chromosomal inversions, as I was saying. So if position A1 to A2 matches with position A2 to A1 here. So you'll have these offset diagonal lines which are going to be perpendicular to your leading diagonal. So this will tell you that there's an inverted sequence match between protein A versus protein B or gene A versus gene B. So if you do it for the whole genome, you see very interesting patterns across the genome. So I'm talking interchangeably about genome and proteins. Please don't get confused. And if you're getting confused, please let me know. Because the examples that I have are for genomic regions, but I hope you can appreciate that this will be the same for proteins. Right. So if you do it if for the genome, let's say, you will see very interesting patterns across the genome. So, for example, this is the one. Uh, so, this suggests that large chromosomal inversions reverse the genomic sequence symmetrically around the origin of replication. Such symmetrical inversions appear to be a common feature of bacterial genome evolution. So, this kind of patterns inside the genome can tell you a lot of things, including evolutionary constraints as well. So these are very interesting things that you can find by simply doing a dot plot. Right. Okay. Okay, now when we talk about alignment of two sequences, so by dot plot, we are just trying to see how similar two sequences are in a very abstract manner. There is no scoring involved, there is no penalties involved, there's nothing involved. Just want to see if we have a more or less approximate match of two set of sequences or not. But if you want to find the good alignment, so if you want to have a good idea about how much of sequence 1 is aligning or matching to sequence 2, and how much of it is not matching. If you want a good idea of that, we need to do sequence alignment, right? So what we do is basically we write one sequence along the other so that to expose any similarity between the sequences, each element of a sequence is either placed alongside the corresponding element in the sequence or alongside a special gap character, right? We'll come to that. So for example, let us take these two sequences. So you can imagine these are single letter amino acids, and these are like two set of proteins that you're given to align. Now, you can just take these two set of sequences and just align randomly by the way you want, and you can do an alignment like this. So here the GG match, the K matches, and the G match again here, and this does not match here, and there is a gap here. Right? But the point is, if you have randomly done this, is this the best alignment possible, or are there better alignments that can be done using these two sequences? How can we compare the goodness of two alignments? So if you want to do that, if you because if you sequence A can match to sequence B in n number of ways. If you want to see which is the best alignment, you need to have some scoring patterns, right? So if you have, say, for example, 20 alignments, if you give scores to all these 20 alignments and say that I pick the best alignment based on the most highest score. So that is one way to do it, right? Because other than if you don't have a scoring pattern, you cannot differentiate if one alignment is better than the other or not. So for that, we have a scoring pattern, and we have a way of computing the maximum score of the alignment as well. So how do we like think of the scoring patterns, right? So I am going to again use genomics and genomic sequences and protein sequences interchangeably, but know that the idea is same in both the cases. Say for example, you have X and XY, which are two aligned pair of elements of two sequences. Not X and Y are not sequences. X and Y are individual elements of those sequences. And if X and Y are same, you put a 1. And if X and Y are 0, X and Y are not matching, they're not the same, you put a 0. So that can be one form of scoring. Right? So the score of an alignment would be sum of scores of the identical pair. So if you just do an alignment like this, the first one does not match, the second one matches, third one matches, this does not match, and the third, this one matches again. So we have three elements matching out of five, which is like 60% identical. I think, I thought somebody said something. Okay. Okay, now this thing, this gap that you have introduced, right? So what does the gap mean in the biological system? And in say, for example, you are aligning this a particular genomic region to another genomic region, and you see it matches almost entirely, but there is a gap there. What does this gap mean? Biologically, it means,
Uh,
yeah.
Indels.
Yeah, biologically it
Indels.
means indels, right? So there are some portion of the DNA might have been deleted off, or some portion of the DNA might have been inserted, and so on and so forth, right? So these actually the insertion and deletion of this small set of sequences inside the DNA, this is not very trivial. You just cannot like randomly insert or delete sequences in a DNA. The cell or the organism does not, nature does not allow that. Nature only allows that when there is an evolutionary benefit from that mutation across certain generations. If there is an evolutionary benefit from that particular gap or the indel will remain in the genome for multiple generations to come. If there is no evolutionary benefit, or if it is costing you, if it is producing some disease or something like that, it will be purged out, because that if say for example there is an indel inside the region of a very important gene of an individual, and that is actually causing a disease to that individual, so that individual might not be able to reproduce and pass on that particular mutation to the next generation. So evolutionary wise, that particular mutation or indel will be purged out of the population, and if it's a evolutionarily beneficial mutation, it will stay. So these gaps, these are not just gaps in alignments. These have some meanings. These have evolutionary meanings. These have very important connotations, right? So if you're just introducing a gap inside an alignment and you just take the score to be zero, that is not very well representation of how the biology is actually working. So in order to introduce a gap inside a genome or a protein or whatever you're aligning, that has to be there has to be some reason to it. There has to be some evolutionary benefit to it. So or constraint to it, depending on like if it is harmful or not harmful or whatever. So that has to be properly represented in your sequence matching algorithm as well. So you cannot just introduce a gap. There has to be some cost to introducing a gap inside the genome. So for that, what they do is uh, uh, they correct this by introducing a gap penalty. So if you are doing a sequence alignment and if you want to introduce a gap inside that alignment, then you have to add a gap penalty, gap opening penalty for that. And how many number of sequences you are, how many number of elements you are using in the gap. So if you are using say 10 elements in the gap, you have to have a penalty for those 10 elements and also a gap opening penalty. So it is given as the gap opening penalty plus the length of the gap uh, that you are inserting there or deleting from that region. So for that we have these two things. So gap opening initiation and elongation. For both of these things for gaps, you need to add penalties for them. Uh, okay, I thought. Uh, there are raised hands. So yeah, please go ahead, Lakshman.
Sir,
Okay. Yeah, yeah, go ahead, go ahead.
sir, you talked about dot plot. So this is a part of a dot plot, or it is another
No,
way of alignment?
no, so it is not exactly, it's not exactly either. So dot plot, what we were doing in dot plot, just let me go back there. This is not a good example. Yeah. In dot plot, we are just, it's a very simple thing. If there is a match, we put a dot. If there is a match, we put a dot. So the idea is that there is no penalty, no alignment, nothing. We just want to visually see if this sequence and this sequence are somewhat similar to each other or not. There is no quantification. Is exactly how much similar. Is it like 100% similar? Is it 80% similar? Is it 60%? We don't know about that. We just want to see visually, is there a symmetric similarity there or not. So that is the use of a dot plot. Dot plot is a very preliminary way of seeing if two sequences are similar or not. But if you want to quantify it, how similar is it and how different is it, then you need something like sequence alignment where you have a scoring scheme. The scoring scheme is the most important thing of the sequence alignment part, that you have certain set of scores assigned to tell you if the 20 alignments that you're doing, which is the best alignment out of that. That is the purpose of sequence alignment. So for dot plot, it is just to
Okay, sir.
see visually, and for sequence alignment, you are quantifying the similarity between two sequences. Okay.
Okay, sir.
Yeah. There's another question, Lakshman. Yeah, go ahead. Are you saying something? You're muted. Okay. Uh, you can write in the chat box later if you want. Okay, let's move on. Yeah. So as I was saying, so if you have a gap, you need to penalize that gap properly. So for opening a gap, you need to have a penalty, and extending the gap, you need to have a penalty. Right? Okay. Uh, just a second. Yeah. Now, what we were doing here is basically identity scores. So basically, if two elements are exactly identical, we put a one, and if not, we put a zero. And with gaps, we add some penalties, some negative scoring to the alignment. Excuse me. But there's a problem with this identity scores. So if in two pairs of aligned sequence there are mutations in the sixth position, you can see these two examples, position, and insertion or deletion on the fourth position. So these two are uh, so I think this is this particular T G K. No, these are different. Okay. Uh, there are mutations in the first and the sixth position. So the first position is a mismatch, then GK matches, then there is a gap here, then G is again matching, and the sixth position there is another mismatch. However, at the first and sixth position, and insertion or deletion in the fourth position. However, V and A share a significant biophysical similarity, and we often see mutations between them. So now here comes the caveat. The biological information is that even though V and A is a mismatch, right? V and A, uh, although there is a mismatch, but chemically V and A are very similar in each other, like biophysically and chemically, both of in a physical structure, in a protein structure, if you put a V instead of an A, the structure is not that dissimilar from the original structure, or the original function is not fully lost in the mutated protein, right? Because those two amino acids are very similar in nature in terms of biophysical properties, and we see substitutions between V and A very often. But on the other hand, if you have a W here, the mutation between W and A is very low. We do not see this happening at all, and they are chemically and biophysically very different to each other. Now, what if you mutated to V and then back to I? Should this have the same score as when I is unchanged, right? So the idea is, let's not complicate this. If a V versus A is zero, should W versus A be also zero? Because both of them are not the same thing. Because V and A in a biological sense are not very dissimilar. W and A in a biological sense is very dissimilar. So we need to have specific scorings between. So if scoring systems for specific mutations. So if you have a W versus A, you should have a different score. If you have a V versus A, you should have a different score, because biologically those two mean very different things. Right. I think somebody. Yeah, Lakshman, go ahead. Did you raise a hand? Okay. Okay. I have another question here. Sometimes after doing the assembly of sequence, we get 1,000. Sorry, Pooja. I'm not very sure what you are talking about. So can you explain a little bit more? Gap clause. I have not heard of this. I don't know what this is. Uh, I think this is slightly different.
Sir, how do we know that there are number of ways, how many number of ways that we can align a sequence?
Sorry, can you
Sir,
repeat?
for example, we have two sequence. Sir, for example, we have two sequence
Yeah.
A and B. So how do we know that how many ways we can align? Is there any formula to know how many ways we can
No,
align this
you
sequence?
have to do it in all possible ways. So if there are, so for example, there are four, um, uh, it's a sequence of four, and you're aligning with another sequence of four. So it can be four times four, 16 alignments can be possible there. So ideally, you would do 16 alignments, score each of the alignments, and take the best possible alignment. That is the
No, sir,
ideal way.
sir, it's just like we just multiply it by two. Matlab, for example, I have eight and a sequence of eight and a sequence of six, so I can have
It's
48 alignments.
Yeah, you can have 48 combinations of these alignments. Now you'll have scores of
O-
48 alignment situations. You choose the score with you choose the alignment with the best score.
Okay, sir.
Yeah. So technically that is how it is done, but computationally it is very intensive. So if you use computational softwares, most of them will not perform all these possible alignments. They'll do heuristics and they'll try to reduce the number of possible alignments. And there can be you can use different techniques to do that, but that is to reduce the computational load of the software itself. But technically speaking, you should do all combinations and take the highest possible one.
Okay, sir.
Yeah. So this question we get no. I think Pooja. I'm not very familiar with this particular software, but from what I understand, it's basically uh, it's not exactly, I mean, this is alignment, but this is not direct alignment. I think this actually is, you're talking about assembly. So this is actually, I think there will be an assembly tool that does the assembly, and this particular tool actually improves on that assembly, if I'm not wrong. I'm not very familiar with this particular thing, but I am not very sure what exactly you are asking from this. But this is not exactly an alignment thing. So assembly and alignment are different things. So assembly is basically you do not know what your final sequence should look like, and you have a set of smaller subsequences of that sequence, and you try to combine these smaller sequence to make the larger sequence without any a priori knowledge of the larger sequence itself. That is assembly. In alignment, you have a known protein or a known DNA segment or whatever. So you have a known sequence and you're trying to align an unknown sequence to the known sequence. So that is alignment, and the scores is based on how much similarity the particular sequence you're trying to align is similar to the original sequence. So that is alignment, and assembly and alignment are different. Just to make it clear, and I don't think here that thing is happening. Here it's more on assembly, and it's not even, I don't think it's an assembler. It's actually works on top of an assembler, if I'm not wrong. So I'm not very sure of this particular tool, but yeah. Can you explain what do you mean by this? That we close a gap and we lead to a valuable sequence?
Sir, actually, suppose when we are, if we are ignoring that gap and like we are closing the gap, sir, we are saying that, right?
No, we are not closing the gap. What we are doing is basically, so just let me go there. Yeah, say for example here. So say all these possible combinations. So what we are doing is, in this case, we are introducing a gap so that the alignment of the rest of the sequences is good. So is introducing a gap in the alignment leading to a better alignment or not, is what we are trying to do here. Uh, I'm not sure what do you mean by closing a gap.
So sometimes, like when data which is generated from DNA sequencing methods and all,
Yeah.
so there they give the sequence. We are
Yes.
getting the sequence,
Yes.
but there I have seen that like they give the gap, and then also sometimes they put the signature sign of indels.
Yeah. So, are you talking about, say, for example, an IGV kind of a thing? Or, like, see, the thing is, there also sequence alignment is happening. So, so for DNA sequence analysis, what they do is they chop your DNA into smaller pieces and overlapping pieces. And now these pieces are sequenced. And after sequencing, you have to know where these pieces of DNA actually came from your original genome, right? You do not have that information. So for that, you have a reference genome. So in for humans, that is GRCS37 or GRCS38 or T2T, whatever, you have a reference genome with the full sequence, full known sequence, 3 billion base pairs of the human genome, and you take the short reads that you have generated and try to align to the whole genome and see where it is aligning. So while aligning, you will be computing different scores for aligning this particular sequence to different regions of the genome, right? And ideally, if the score will be maximum for the region where the read has originated from, so that you can have a good alignment at that particular. So your alignment is correct. So that is how the
Okay,
DNA
okay.
yeah works, right? So yeah, there
So
are
that means I will be considering the so that means I'll be considering that sequence and I will be taking the small part of the sequences and then again when I'll be aligning and getting the best code that will be giving me the best result.
yes. Technically, huh. Technically, if you're given a base pair of 300 base pair long read, like in
Hmm.
a very simplistic manner, you would try to match that 300 base pair all across the 3 billion long genome of a human individual. You have that 3 billion long sequence, and you have a sequence of
Hmm.
around 300 base pair, and you're trying to see where does this 300 base pair occur in the genome, and you'll try different places in the genome. You'll have different alignment scores for that alignment.
Hmm.
You pick the one which has the best alignment score.
Yeah, I got it, sir. Thank you, sir.
Yep. Okay. Uh, any more questions? Yeah, in most of the cases, it is confirming the presence of a large mutation. So generally, there are mutations in the genome called structural variations, right? So those are variations which are more than 50 base pairs in length. So it can happen in due to various biological reasons. A significant region of your genome has been deleted. So we see like 500 kb or like say 200 kb or 100 kb base pair length DNA sequences have just been deleted off from the genome of that individual itself. Now, when you align the genome of that individual to a reference sequence, you'll find a 100 kb base pair gap in the alignment. So that alignment will tell you that there is no data in that region from the individual that you have sequenced. So that means that individual does not have that sequence in their genome. So that is a deletion that has occurred in that genome. So it technically tells you of these large mutations. Even small mutations, these gaps can be like of like 4 or 5 base pairs as well. But those are also mutations, small indels that has happened, mutations that has happened in the genome of that individual you are trying to study. Yeah. Okay. Uh, yeah. As I was saying, the substitution, I mean, so the mismatch is not the same in all cases. So a V versus A mismatch is not the same as a W versus A mismatch. V versus A mismatch is much more probable, much more evolutionary constraint probable. Like it is not. it is very widely seen. Like in a lot of individuals, you can see this particular mutation and they're surviving fine. So there is no issue with that particular substitution, although that is a mismatch. But in this case, a W2A mutation is very rare. You won't find it. And if it is there, there is some malformation of the protein, some disease that is getting caused or something of that manner. So this alignment is so this mutation is much more harmful, dangerous, or you may call important than this particular mutation. And your alignment scores, the scoring system that you are using for aligning these two sequence must keep in mind of this kind of things, that all substitutions are not the same. Right. So for those kind of things, we have scoring matrices. So I think Sir has discussed PAM and Blossom. I'll just quickly give you an overview of what these things mean, but I'm not going to go into very details. But I hope I have given you the idea about why we need these scoring matrices, right? So that is more important than learning the matrix itself. So an amino acid scoring matrix is basically a 20 by 20 table. So you have 20 possible amino acids. So it's a matrix of 20 by 20 dimensions, and each element inside the matrix will tell you about the score. If A is substituted by W, what is the score? When W is substituted by S, what is the score? So you create a matrix like this, which is predefined. So if you can have an identity matrix where basically exact matches receive one score and non-exact matches receive a different score, or you can have mutation data matrix, which takes things like this into account, that what kind of mutation is happening, and according to that, what is the score of that. So in mutation data matrix, it is a scoring matrix compiled based on observation of protein mutation rates. As I was saying, V2A is a much more frequent mutation that is seen, but W2A mutation is not seen. So for this mismatch, you'll have a certain score, a certain negative score, but in for this particular mismatch, you'll have a higher negative score to penalize more. Right. Uh, so yeah, you can construct a scoring matrix compiled based on observation of protein mutation rates, and depending on some mutations are observed more often than other, and you have matrices like PAM and Blossom to help you with this. Uh, so yeah, the scoring can be based on different things. It can be based on biophysical properties. As I was saying, V and A are very similar. So you'll the mutation score, the mismatch score of that or the penalty for that will be very low. And W versus A is a much different mutation and a much higher concerning mutation. So you have a higher score, higher negative score, or a higher penalty for that particular substitution. Right. I think I'm going to skip this. Okay, just let me quickly tell this. So this is in case of DNA. So similar in case of proteins, but I'm just giving an example in terms of DNA. So you have these four base pairs, right? A, G, T, and C. So conversion of A to T is basically transversion. Conversion of A to G is basically transition. So we have purine to purine as transitions and purine to pyrimidine as transversions, right? So, and if you can calculate the probability of this transition and transversions, the probability of these things happening in a certain unit of time, right? So TAM is basically that unit, which is percent accepted mutation, which is the time needed to acquire one mutation per 100 positions. So one mutation per 100 positions, what is the probability of that particular substitution happening? So you have a probabilistic score for that. So if you assume equal probability, so each for equal probability, each mutation, if you assume a PAM matrix like this, you get something like this, which is alpha. Alpha and beta are basically the probability rates of transitions and transversions, and you get a probability rate of something like this. And in some cases, in some other people have uh, proposed models which take into account different kind of scorings between purines and pyrimidines, and you have probabilities of something like this. So you can either have equal probability, so your transition and transversion rates are equal, which is like 0.3%, 0.33%. And in this case, the transitions and transversion rates are not equal. The transversion rates are slightly higher than transition rates. So these are like based on predefined data, predefined understanding of the mutations that are happening, how frequent they are, how harmful they are, and so on. Right. So that is important. So, yeah, I'll not go into very details, just quickly I'll go over this. So, if you consider a probability of observing amino acid B as PB, so if you do M of AB, so the mutation of AB, odds of seeing that substitution by PAPB, which is the probability of observing amino acid A times probability of observing amino acid B, which is basically the odds of seeing substitution as a result of mutation. So if you calculate this as the odds, uh, and if you want to calculate the odds of seeing the whole alignment by chance versus a result of mutation, it is given as a product of this thing in a log format. I'm not going into the mathematical details, but in this predefined scoring matrices like PAM and Blossom, the scoring function looks something like this. This is how the score is calculated based on probabilistic values. And PAM is basically, as I was saying, point accepted mutation or percent accepted mutation. And two sequences are defined to be one PAM unit diverged if a series of accepted point mutations, no insertion deletions, basically no gaps, can convert S to T with an average of one mutation per 100 residues. So if you have a sequence S and a sequence T, if you do one mutation per 100 residues in S, can you convert that S sequence to a T sequence? If that is possible, that is basically one PAM unit, as they say it. Right? Uh, and it is possible for two sequences to be multiple hundreds of PAM mutations apart because one position
Sir,
can. Yeah, yeah.
sir, can you re-explain the log score? My connection got lost a little bit.
Okay, it's quite simplistic. So MAB is basically the probability of this particular mutation happening A to B, and PAPB is the probability of observing amino acid B at that position times the probability of observing amino acid A at that position. This is the score. This is the odds that is defined for the substitution at that position for that particular substitution, which is A to B. Now, if you take log of this whole odds ratio, which is the log odds, if you take a log of that ratio, that is what the score is that is present in a matrix like PAM or Blossom. So the score is basically a probabilistic value of seeing that particular variation at that particular position in the protein or the genome. So it's
Sir, how do we calculate PA and PB or MAB?
for that you need data from data from sequence of hundreds of individuals hundreds of individuals. So you can study what is the probability that in a set of 1,000 individuals you're seeing this mutation. What is the probability of that? In a set of 1,000 individuals, you're seeing mutation B. What is the probability of that? So you need a lot of data and lot of understanding of what is happening to kind of create this particular matrix. As I was saying, PAM and Blossom are basically predefined matrix. There has already been studies that has calculated these probabilities using different statistical models, and they have incorporated those as scores in the PAM and the Blossom matrices. Yeah.
So, sir, if I were to align two sequels, so I can look the scoring at PAM
Yes,
and Blossom.
yes. Those matrices will tell you
Okay,
what
sir.
should be the score if one particular substitution has happened.
So they are just
Okay, sir.
reference, right?
Thank you, sir.
Sorry?
That means they are just reference matrices, sir.
Yeah, they are reference matrices which has already been pre-calculated based on prior data and whatever we have seen in the particular organism. Yeah,
Okay, sir. Thank you, sir.
yeah.
Thank you, sir.
Okay. Last set is basically a little bit of enzymes, and in terms of like uh, since enzymes are very important, the most important proteins of all. So how do we How do we kind of identify enzymes using different identifiers? So there is this enzyme commission, which has created this number system, and it works like with the Dewey Decimal System or the Library of Congress classification, but it is for enzymes. So each enzyme is assigned a unique four-part EC number based on the type of reaction it catalyzes. So these are the major seven type of enzymes that are classified for organisms. So you have oxidoreductases, you have transferases, hydrolases, lyases, isomerase, ligase, and translocase. Right, and these are the type of reactions these particular enzymes catalyze, and these are the EC numbers. So EC 1, 2, 3, 4, 5, 6, 7, so seven different classifications. These are the types of reactions, and it catalyzes. And these are the trivial names for that. So if you see a dehydrogenase, it means that it is removing a hydrogen ion from the compound. So it is of the class of oxidoreductase. It can be a kinase, which is transferring a phosphate group to a particular protein. That is basically transferase because it's just transferring the group, and so on and so forth. So these are your trivial names. I think you have seen a lot of these, and these are their class names, right? And these are their basically the class numbers for that. And so basically the EC number is for a particular enzyme is a four part number, and they're separated by periods, which are basically full stops or dots, right? So the first digit will tell you the enzyme class. So if it is a 3, it's a hydrolase, as you are seeing here. Enzyme class 3 is a hydrolase. If it's a 3.4, it's another subclassification. Second digit is basically the subclass of the first one, which is the specifies the type of reaction within the main class, often based on the type of bond or the chemical group that is involved. So hydrolase meaning it is breaking hydrogen bonds, and it is basically bringing the water out of the molecule. So that is a hydrolase. Then you have 3.4.1 is peptidase that cleave the first amino acid of the polypeptide. So the third digit will tell you the reaction mechanism or the specific type of chemical transformation that is happening. And the fourth digit finally will tell you uniquely which particular enzyme is that. And in this case, is basically 3.4.11.4, which is an enzyme that removes the first amino acid from a tripeptide. Right? Sorry, there is one more question that I saw. Can there be variation in sequence between two? Yeah, so this is a very good question, and this is a question that I try to kind of look at my research work. So yeah, two random individuals, if you take from any part of the world, two random human beings will have 99.9% sequence similarity across their genome. The 0.1% dissimilarity that is present in the genome makes us who we are, gives us our unique identification, unique characteristics, our hair color, our eye color, our height, our other things basically, which are genetically controlled. So these are what makes us unique. Our kind of risk to different diseases. All of these things are conferred by that. But other than that, 99.9% of the sequence is similar between any two random human individuals. Now, there can be variation in sequences between two human beings in a particular gene, as you have said, salivary amylase. It can be possible. Say, for example, a healthy individual has a normal salivary amylase gene, and another individual who is not healthy, has some disease, might have a deletion in that particular gene. As I was saying, these big structural variations, it might happen that that individual's DNA has been mutated, and in that particular gene, a certain number of base pairs have been deleted. So that is possible. But generally, what this will lead to is some sort of disease phenotype in the mutated individual if it's a very important gene. But if it is happening inside non-coding regions, in intronic regions, intergenic regions, it might not be phenotypically visible. It not might not cause a disease or something else. It can cause a disease. It's not like if you have variations in your intronic regions, it won't cause diseases. That is not true. It can cause diseases, but in most cases it won't. And so yeah, so any two individuals might be different in many uh, regions of their DNA, but where the difference is will tell you how different the phenotypes of two individuals will be. So if it is in a coding region of a very important gene, you might see phenotypic differences, but if it's in intronic, intergenic, 3 prime, 5 prime UTR regions, possibly you won't see any differences in individuals. Yeah, sickle cell anemia, on the other hand, is basically, I was talking about structural variants. Sickle cell anemia is a missense variant. It's a single nucleotide change in the HBB gene of individuals. So there's one single nucleotide change which changes the amino acid sequence of the hemoglobin protein as well. So the hemoglobin protein becomes sickle shaped rather than a normal shaped hemoglobin protein. So that is also like if between two individuals, one has sickle cell anemia and another is a healthy individual, you'll only find one mutation, one point of difference in the HBB gene, which is causing this particular phenotype to happen. Yeah, I hope that answers your question.
Yes, sir.
Yeah. Okay, then yeah, in terms of the recap part, I'm done. If you have any questions, you can ask, or else we'll move on to the uh, the assignment sections. Okay.
Sir, if we
Yeah.
compare, sir, if we compare sequence of two healthy individuals, like
Yes.
healthy individuals, so there would be changes in intron region or in extron expressing DNA sequence
Uh,
region.
there can be both. There can be differences in intronic regions. There can be differences in exonic regions as well. But as you know, that all mutations are not directly contributing to any differences in gene expression or differences in uh, mm, protein formation or anything else. So the mutation so if you compare two sequences between a healthy and a diseased individual, the diseased in sorry. That was your question, right? Healthy and a diseased individual.
No, sir, I was just talking about two healthy individuals
Haha,
that
too healthy. Yeah, yeah, sorry.
have
Yeah.
no phenotypic.
Yeah, that is possible that both of them are healthy, both of them do not have any diseases, but they might have differences, some differences in their coding sequences. That is possible because not all of these differences will actually be a factor in downstream processes. Not even if you have a change, if you have a synonymous variant. The synonymous variant is basically it changes a nucleotide in the in the coding region of the individual. But since if you remember our genetic code is degenerate, so you have 64 codons coding for 20 amino acids. So multiple codons can code for a single amino acid, right? So if your mutation happens, like you move from one codon to another codon in that particular region, but the protein coded by both the codons are the same. Amino acid coded by both the codons are the same. So you won't see any downstream changes. So these are called synonymous variations. So these can be present in the coding regions, but they do not change the amino acids downstream. They do not change the protein structure at all. So there can be differences. There can be synonymous differences. There can be other differences as well, which do not directly contribute to gene expression or protein expression differences. That is also possible.
Sir,
Okay,
like
thank you, sir.
we know that the two individuals differ in the VNTRs, right?
Sorry, you are not audible. Hello.
Sir, we know that two individuals vary in the VNTR regions, right, sir?
That
Variable
number of
numeric.
yeah. There are repetitive regions in the genome which are basically tandem repeats, and VNTR stands for variable number of tandem repeats. It is possible you have a lot of different. So you have a repeat subunits. If a repeat subunit of 5 nucleotide which has been repeated 500 times in one individuals, and say 480 times in another individual, 520 times in another individuals, that is possible. But most of these VNTR regions are non-coding regions. So anyway, from a protein coding point of view, these might not contribute
Not
that
very
much into,
valuable.
yeah.
So we don't get much information from the VNTRs.
We get, it's not that we do not get, it tells us a lot of things, but in most cases, VNTRs, the differences in VNTRs will not result in a difference in phenotypes or healthy versus diseased individuals. But there are, I'll give you another example. So, if you have heard of the disease called Fragile X syndrome, right? So, Fragile X syndrome is a very rare disease that happens when there is a repeat in the FMR1 gene in the X chromosome of the individual. So what happens is it's already a very repetitive region, but the number of repeats increase beyond a certain threshold, and after that, this forms a disease. But if the repeat number is below a certain threshold, the variation is not pathogenic. But it after it reaches a certain threshold and it crosses that, it becomes pathogenic and you form the disease. Right. So the number of repeat units matter in certain cases. If it is in within coding regions like the FMR1 gene, like Fragile X syndrome, you will see a diseased phenotype. But in most cases, these are in intronic regions. In most cases, these variabilities are in intronic regions, and in most cases, you do not see a effect of that.
Oh, thank you, sir.
Yeah. Uh,
So,
yeah.
I heard that nowadays, mainly I heard in US, it's very common to get your genome sequence to see if we carry any disease or we can expect any disease in the
Yeah.
future.
Yeah. Yeah.
So, how is it analyzed, sir?
So that's a discipline in itself, analyzing genomic sequence data. But I'll tell you shortly, it's basically, as I was saying, that you sequence the DNA in very small stretches. You cannot sequence the DNA end to end 3 billion base pairs long. What you do is you break it down into smaller pieces of like 500 base pairs, and then you sequence those 500 base pairs. And after you're sequenced, you have this 500 base pair sequence, but you do not know from where in the genome this originated. For that, you do sequence alignment and you find the best possible alignment of those particular reads that you have generated from sequencing, small reads, to the big reference genome. After you have aligned, in the alignment, it will not be a perfect alignment. It will not be 100% alignment. There will be some bases that are not matching inside those alignments. So if those mismatches are statistically significant, we use a variant caller for that, which tests the statistical probability of that particular variant, that particular position to be of a different not amino acid, a different nucleotide. Then we call that particular uh, location as a variant. So, say for example, you have 100 reads that is supporting that. So you have total 100 reads in a particular position A, right? And 50 of those reads are telling you that at that position there is a C, and 50 of the rest of the reads are telling you there is another, 50 are telling you C and 50 are telling you there is a T, right? So, since we are diploid organisms, we have two copies of the same DNA, right? So, what it means is 50% of the DNA is having A at that position, and 50% of the DNA is having T at that position, which means it's a heterozygous variant there. So one allele is A and the other allele is T. It has been mutated. So depending on the alignment and after alignment, what is the base that is supported by the alignment, we call variance. We say that with this much probability or this much certainty, I can say that there is a heterozygous variant in this region, there's a homozygous variant in that region, and so on and so forth. So while DNA testing, what you do in DNA testing is basically sequence and DNA and try to find variations, and after you have found variations, see if these variations are known to be causing diseases. Because there is a lot of prior literature that has studied this set of variations. There are databases like ClinVar, like Intervar, and other HGMD databases which tell you if that particular variant is known to be causing some disease or not. So generally, this is how it works. This is for smaller variants. For larger variants, as I was saying, you'll see a sequence that has been deleted off, and so on and so forth. You make predictions on if that will affect your genome and can cause diseases and so on and so forth. Like that. Yeah. Okay. Uh, any more questions? Prokaryotes, I'm not very sure. I have never worked with prokaryotes. I'm not very comfortable also. Like I've not handled any bacterial genome data or anything like that. So I might need to look at. So uh, I'll try to find one. I'll keep this as a note, and next time I'll try to find one. Yeah. Okay. Okay, then we'll try to solve the questions. Hmm. What of these? Okay. Yeah. So again, I have not gone through this, but please check out which amino acids are hydrophobic, which are hydrophilic, which are aromatic. These are like the basic amino acid properties that you should know. Uh,
Sir, I have a question regarding proline. I had post a doubt on forum also, but it has not been answered. So the proline has been listed by Professor Michael in his one of his lectures as hydrophilic. However, when I look up at standard Lehinger, proline was listed up as hydrophobic amino acid.
I think also proline is a hydrophobic amino acid. I'm not sure why professor has called it hydrophilic. And I do not have access to the discussion forums because I think there is a separate TA who handles those things. I am just here for the live session. I'm not very sure about this, but since you have posted it in discussion forum, I think somebody will answer that because there are other people who handle that. So I think maybe, yeah, I'm not sure.
Sir, I for now I should consider proline as hydrophobic
Yeah,
as
yeah,
per
yeah,
Lehinger
yeah,
standard.
yeah, yeah. I think it is hydrophobic. Yeah, and Lenin says hydrophobic as well, right?
Yes, sir, yes, sir.
Yeah, yeah. So then definitely it's hydrophobic. Yeah. Okay. Yeah. So I just wanted to I wrote these explanations on my by myself. So again, like just wanted to tell you what in terms of hydrophobic and hydrophilic, basically uh, isoleucine. Where is isoleucine? Uh, yeah. So you have this long hydrocarbon chain. So any compound that has this long set of hydrocarbon chain are hydrophobic in nature because these do not these just carbon hydrogen bonds, they are not polar in nature, right? They're very stable bonds and they are not polar in nature, so they do not they're so in simple words, these are hydrophobic because they have these long carbon hydrogen bonds, a lot of them possible. So if you have read about cell biology, the cell membrane also has this hydrophobic hydrophilic nature. You have these long hydrophobic tails of these phospholipids that are pointing inside towards the membrane because they're hydrophobic in nature and because of those long tails of carbon hydrogens. So yeah, the reason behind them being hydrophobic is because of the presence of those long non-polar side chains. If you had a polar side chain, it would have been water soluble. This is very simple. C, a single C. Yeah, I'm not going to dwell on this. It's basically a period is what it is. So yeah, okay, yeah. Yeah, first of all, the answer is correct. I just wanted to ask, are these questions exactly the same as you have done in the assignments of week two, or are these slightly different? Or week one even, like last week what we did. Are these like exact same questions? Because I don't have access to the questions of this year.
No, sir, they're slightly different.
Okay, okay, okay. ठीक है. This is something that if you know, you know. If you don't know, I don't think. Answer. Okay, I see a lot of D's. Yeah, the answer is correct. Thrombin is a protease and it converts fibrinogen to fibrin, which actually forms the mesh-like structures, which prevents the blood from flowing out, which is actually the clot. So the fibrinogen is soluble and the fibrin is insoluble, which forms the mesh and forms the clot. So yeah, thrombin is kind of very important. There is another version of thrombin which is called prothrombin, from which thrombin is derived as well. So, okay, somebody answered B. For B, you do not need active transport. B is basically you're moving from higher to lower concentration, which is by default it will happen. You don't need energy for that. So that is passive transport, right? If you need energy for something, that becomes active, right? So active transport is where you require energy, and where would you require energy? Going from a lower to a higher concentration, because you're pushing against the flow. So there you would need energy, and so that is active transport. Generally, ATP is used for this. So you move from a region of lower to a region of higher concentration. So yeah, if you if you are interested in learning active transport or transport of any kind, the most like interesting example I would suggest is the sodium potassium pump and how it works in neuronal conduction in the central nervous system, how it like generates action potential due to active transport. It's a very interesting part of like transport systems and all. So if you really want to understand this, I would suggest learning about sodium potassium pumps and neuronal transport. This I will not dwell time on. This is basically Brenda. We have not directly covered it, but if you have any questions regarding this, you may tell me. I also like I am familiar with the database, but I have not been very hands-on with the database because Brenda is uh, like it is not obsolete, but like yeah, I do not use that much. So that is why I'm not covering. But if you have any questions regarding to this, please let me know. So answer is Brenda, by the way. And yeah, so this, sorry. Uh, yeah. Hi, Lakshman, you wanted to ask something? You can unmute and ask. Your hand is still raised. Okay. Yeah, we discussed this. It's 1 in 100 residues. That's correct. Let's see a lot of E's. Okay. Yeah, the answer is correct. Although I have not discussed why. Um, there are a few reasons why. Firstly, because if you have a small number of residues, it and like 3 to 4 residues or 4, 5 residues are enough for specificity with binding with the substrate itself. So you don't need like 500 residues for specific binding. 3 to 4, 5 residues are good enough. So you are saving energy on that front. Uh, Also, it is conformationally very flexible in terms of that uh, the enzyme shape change can be induced very easily if you have a very small number of residues. Uh, so if you have, meaning like these small sites will allow for a mechanism of induced fit of the enzyme, which is basically the enzyme will change the shape slightly upon binding with the substrate. So that thing, conformational change, is also very important, and it is flexible if you have a small number of residues which is binding. So yeah, there can be other reasons as well, but this is also a very important, interesting topic that you can read about. I think there are a couple of papers which talk about this, like why do we have like such a small number of residues for such a big protein. So yeah, this is interesting as well. Yeah, that's true. It also limits the number of interaction. Yeah, that's true. Yeah, the answer is all of the above. It is dependent upon what question you are trying to answer. Are you trying to answer, are these two sequences similar or dissimilar because of evolutionary constraints? Are these two sequences similar or dissimilar because the amino acid residues are completely different in nature? So, the type of question that you are trying to answer will determine what kind of scoring matrices you use. But you can generate a scoring matrix using any of the properties of the amino acids. Yeah, this also we discussed, I think. Yeah, yeah. So you have a gap opening penalty and you have a gap extension penalty. Generally, it is given like this: gap opening plus length time gap extension. So yeah, that's correct. So yeah, that's all from my side. Do you have any questions?
Sir, I saw a YouTube video on a research happening in US regarding to bring back woolly mammoths using elephants' DNA and mammoths' DNA. They
Yeah.
have already they have already formed a transgenic woolly mouse, and they are doing so. That's I found it interesting that that's happening in the world.
Yeah, I think they have already gotten that dire wolf. So if you have seen
Yes,
Game
sir.
of
Yeah. Oh, yeah,
Thrones,
wolf
yeah,
also. Yeah,
yeah. So they have
yeah,
already
wolf
brought back
also.
dire wolf. There are a couple of issues with that. So they are just changing a few mutations. They're doing a few mutations in the genome of its nearest ancestor and trying to get the extinct animal. But there are a few issues with that. I personally find is basically you are just trying to mutate the genes that give the uh, what do you say, the external look of the animal. So it phenotypically, just the outside skin and how it looks might be very similar to the animal that has been extinct, but it is not the same animal. There are a lot of genetic differences between the original direwolf and the transgenic direwolf that has been created. There are a lot of other ecological issues as well. So you are technically not bringing them back. You're just making them in a lab and like creating them for show, because you cannot, bringing them back would be like putting them in their ecological position in nature, which you are not doing, which is not possible. Even like uh, they have gone extinct millions of years ago. If they are brought back into the same ecological system, they might not survive. Other animals might eat them, and so on and so on. So technically it is a good science experiment to be done, but realistically speaking, it's not the same animal. And secondly, it's uh, just for an experiment. I don't think like it is very difficult to bring back extinct animals if specifically if the animal has been extinct a very long time ago. So I think dire wolf has been extinct a very long time ago. And, like and technically the animal that has been born is not a dire wolf at all. It's a wolf a little bit in disguise. So mm.
Okay, sir.
Where did I lose?
Sir, also what people defy GMO, genetic modified plants, and they do not want to eat. Is that we should
No,
eat or
I
we
don't
should not?
think that is more of a superstitious kind of a thing rather than anything else, because GMOs for plants have led to the green revolution in India, in countries like India where there was food scarcity. So you had GMO crops which yielded more crops, you had better quality food. So plants GMOs is like it's not a problem. Animal GMOs, depends on how you are using it, because there are a lot of ethical concerns when you are genetically modifying an organism, whatever organism that might be. Plants is okay, but animals, it's a ethical issue. Uh, but yeah, but in terms of eating, I don't think that's a problem. Actually, it's in some cases it's better, because by genetically modifying, what they do is they try to increase the nutritional value of that particular plant. They try to increase the yield. They try to increase antimicrobial resistance, and so on and so forth. So in those things, like it is better. But that's more of a philosophical question than a scientific question.
Okay, sir. Thank you, sir.
Uh, how is Yeah, yeah, we are not speculating. It's just that, like, it's not the exact same DNA. If we were talking about sequence alignment, right? So if you take the DNA sequence of an extinct direwolf found in fossils or something like that, and you match the entire sequence to the direwolf that has been created, it's not going to be the same. Some genes are similar. The genes that give the outer look of the animal, those things are similar, but a lot of things are not similar. It's more closer to ancestry wise. If you talk about ancestry, it is more closer to the current wolf than the dire wolf itself. So it's it's like you have not like made the the individual animal exactly like the same it was. It's different. Yeah, so okay, I think that's all for today from my side. I think we'll meet next week, same time. And yeah, if you are the discussion forum thing, if there is no answer on the discussion forum thing, try to mail. I think you might have been given email IDs of TAs and Professor Michael as well, and I think other people from the NPTEL team as well. So try to mail them and tell them that you have not gotten the answer, and like you want the answer for that. And if that doesn't work, like bring the question to me next week, or you can email the question to me as well. I just cannot see the discussion forum because my access is of older NPTEL course, not the current one. So if you still do not get any answer from that, so please write me a mail or bring it up
It's
next week.
fine. I got, sir, it's fine. I discussed it with you and it's okay.
Okay, ठीक है, then good. Thank you for joining. See you next week.
Thank you so much, sir, for this lecture.
Thank you.